I'm Bruce Chapman, and I'm really delighted to welcome you all here tonight, where we are introducing a new book, a subject-defining book by one of the stars of Discovery Institute. It's clearly the most significant step ahead in the debate between Darwinism and intelligent design. I believe that the book and Stephen Meyer are destined to change that debate. Discovery Institute, if I can boast, is home to many successes this year. Some of you may have listened today to Michael Medved, who has probably the most intellectual and stimulating debate format on national radio. And I'm delighted to say that he is a fellow of Discovery Institute. He had Steve Meyer on just recently, and I think he's going to have him back because it's become such a hot topic. Michael, of course, writes a number of books himself. Likewise, another controversy that is appearing in the news is what's going to happen in Iran? What kind of threat is posed to Israel? So we're very excited that at the end of this month, another one of our luminaries at Discovery Institute, George Gilder, who helped found the Institute with me almost 20 years ago, will have a new book out called The Israel Test, which explores that subject in depth, and I can commend it to you. He's another thinker who moves subjects way ahead. In the transportation field, Cascadia Center has done the same thing. In the field of Russia, our Real Russia project has done that. In religious liberty, our program on religion and public life has done so. And especially, I might point out the human rights and bioethics program that is headed by, co-directed by Wesley J. Smith and the former Congressman and Ambassador John R. Miller. But the Discovery project that probably has caused the most attention and garnered the most interest is the Center for Science and Culture, which is headed by Dr. Meyer, and about 45 fellows and staff around the country and even around the world make it work. The Discovery motto is making a positive vision of the future practical, and that certainly is exemplified by this program. So is the Center's own motto, which is exploring the ideals that made Western civilization exceptional. You see, when we talk about a subject like bioethics that gets into so many contemporary issues, you're talking about a question that is posed for our age, which is, what does it mean to be human? And that question takes us back to even more fundamental questions, such as, what is life? What is the meaning of life? Where does it come from? Where do we come from? Now, 15 years ago, when the Center for Science and Culture was first convened, we had a fairly simplistic argument going on in this country and around the world, and that was the argument, the age-old argument between evolution and creation. And yet, when intelligent design came along with a scientific attack on Darwinism, it was launched by Phil Johnson and a number of other scientists in the 90s, the old debate opened up in amazing new ways. In the past year or so, we've seen it open up amazingly even more. It's been underreported, but we have the tactical division among Darwinists. We have the new atheist represented by Richard Dawkins. We have what are called now the accommodationists, who are likewise probably atheist in the implications they derive from Darwinism, but they want to find common ground with people who are of religious bent and accept the full Darwinian case. Then there are the new theistic evolutionists, and they too are divided over the materialist approach, whether it's just limited to biology or you can talk the whole realm of science that falls into that category. There are the scientific structuralists who believe that Darwin was wrong, but that materialism is right, that nature self-organizes. And then there are the people who are what you might call the natural philosophers, who believe in particular that Christians should sit this argument out, that is to say they should deal strictly with the philosophical questions and leave the scientific issues alone. Well, in my view, all of this has served one interesting positive purpose, And that's to explode the idea that this topic can be limited to the old molds, that it can be poured in the old molds and just kept there. In fact, it has become, as it gets out of control, the defining intellectual controversy of our time. And it has the most consequences for our civilization. Now, Stephen C. Meyer, Dr. Meyer, is at the heart of that controversy now, and so is his book, The Signature in the Cell. He was featured in the film Expelled. You saw a little bit of that. He will be featured in a coming film on the Cambrian explosion that is going to premiere, and by the way, we will have a premiere of that next month. His organizing skills over these years has been behind the success of many of his colleagues and their projects. He personally worked in geology in Texas oil fields before he got his PhD at the University of Cambridge in the philosophy of science, where he specialized in life's origins. For 15 years, he's been conducting research as well as directing it. And now that research has produced Signature in the Cell. Our colleague, George Gilder, that I mentioned, advised Steve, put all your best material in this first major book. And he's done it. And it's a winner. Please welcome Dr. Stephen C. Meyer.
Thank you. Thank you very much. It's great to be with you. Thanks for the kind introduction, Bruce, and for the thank you also to the Discovery staff who have done so much to put on this event, and to find that nice little clip with Ben Stein. I had a chance to be in Great Britain earlier in the year, and I was speaking at what I thought was Shrewsbury, but it's actually pronounced Shrewsbury by the British. And it's the place of Darwin's birth. And I was speaking there to give a dissenting voice on the legacy of Darwin. And I found that there's just a tremendous amount of interest in Darwin and his legacy this year. It is the 200th anniversary of Darwin's birth. The 150th anniversary of the origin of species is coming up a little bit later in the year, in November. And Darwin is now so big that he's even on the money. We have the Queen on one side with a 10-pound note and Charles on the back. And so around the world with this anniversary, there is the question about his legacy. What has Darwin left us? What do we know because of Darwin that we didn't know before? And quite typically, the answer to that question is, well, Darwin refuted the design argument. He gave us the idea of evolution. He gave us the understanding of the mechanism of natural selection. But according to many intellectual historians, many biologists, Darwin is principally important because he refuted the old classical argument from design. Francisco Ayala, one of the past presidents of the AAAS, the American Association for the Advancement of Science, puts it this way. He says that the functional design of living organisms and their features would seem to argue for the existence of a designer. It was Darwin's greatest accomplishment to show that the directive organization of living beings can be explained as the result of a natural process, natural selection, acting on random mutations without any need to resort to a creator or any other external agent. Ayala has put it in an even more pithy way, and just recently saying that Darwin gave us design without a designer. Now, that idea is picked up in the works of Richard Dawkins, who is perhaps the foremost spokesman for modern Neo-Darwinism. Dawkins puts it this way in his classic work, The Blind Watchmaker. On the very first page, he says, biology is the study of complicated things that give the appearance of having been designed for a purpose. Now, I'm an old college professor, and it's hard for me not to want to call on people. So if I do, forgive me, but I want to offer a quiz right now for anyone who can come up with the key word in this quotation. It's obviously appearance, right? We've got the Darwinian view is that things look as though they were designed, but they weren't designed because there is a purely undirected, unguided mechanism, namely natural selection acting on random variations, or we would now say mutations, that can produce the appearance or the illusion of design without that mechanism of natural selection being guided or directed in any way. Now, that was Darwin's key idea. Natural selection, of course, being the idea of random variations in a population and differential reproduction. Now, let's put that idea of Darwin's in context. What he was trying to show was that all the living forms that have arisen since the very beginning of life were produced by a purely undirected natural process. And the process, again, is natural selection. Darwin also depicted the history of life as a great tree. And he suggested that all of the living forms that exist today are represented by the branches at the top of the tree. The giraffes, the crocodiles, the birds, the mammals of various kinds, ourselves. And all of those forms eventually could be traced back. They descended by the power of natural selection from originally one or very few simple forms. So that's his theory of evolution, sometimes called biological evolution, because what Darwin was attempting to do was explain all the forms that we have today from simpler pre-existing forms. But Darwin did not answer a fundamental question, a more fundamental question. In fact, he didn't even address it. And that was the question of the origin of the very first life, the form of life that's represented by the very base of the tree, or the trunk there. To get life going, somehow very simple chemicals have to be converted into a living cell. Now, Darwin didn't address that topic, and as we'll see as the lecture proceeds, in 150 years, subsequent generations of scientists who have addressed it haven't been able to solve the mystery of the origin of the first life either. Now, there is question today in the scientific literature and among many of my colleagues at Discovery Institute about the sufficiency of Darwin's theory of getting from that first life to all the living forms we see today. But for the purposes of argument, I'm going to set that question aside in this lecture, as I've done in my book, to focus on this more fundamental question, the question of the origin of the very first life. Can that very first life be explained as the result of a purely undirected process, such that any appearance of design that we might find in, for example, the first living cell can be safely assumed to be an illusion? Or is there evidence of actual design? Is design merely apparent, or is there actual intelligent design? That's the question I'm going to address specifically as it relates to the question of the origin of the first life. Now, in Darwin's time, scientists were not particularly concerned about this. There was a general materialistic turn in science during the late 19th century with an attempt to explain all major phenomena by reference to matter in motion and maybe the laws of nature. But even though there wasn't a formal theory of the origin of the first life, scientists weren't very concerned about not having such a theory, and I'm talking about materialistically oriented evolutionary biologists, because they assumed that the cell was extremely simple. It didn't seem, even though it didn't seem that it was going to be difficult to produce an explanation for the origin of the first cell, because the cell was thought to be something like a blob of jello, which could be produced by a few simple ingredients, a few simple chemical ingredients reacting with each other in one or two simple reactions. I love the quote on the screen behind me because it makes me feel so very smart, being able to look back with the benefit of hindsight and all that's been discovered in the last century, and indeed the last 100 and even just the last 10 years in fields like molecular and cell biology and genomics. But this is Thomas Henry Huxley, Darwin's famous protagonist, his famous bulldog, and he said at the time, the cell is a simple homogeneous globule of plasm. It's a simple glob of jello. So there wasn't a lot of worry about not being able to explain this appearance of design, because scientists at the time didn't think that the cell really appeared all that designed. It just looked like something that a few chemical reactions could produce. They thought that the essence of life was this substance, this goo on the inside of the cell membrane called protoplasm. And all of that changed, first gradually at the turn of the century as we began to learn more about proteins and metabolism, but then dramatically in the 1950s and 1960s during a period of time that historians of science now call the molecular biological revolution. The key discovery in this period of time, or one of the key discoveries was the discovery by Watson and Crick of the structure of the DNA molecule, the famous double helix, with which most of us are very familiar. The double helix is now kind of a cultural icon. We see it on news reports, and we know that criminals are convicted because of DNA evidence, and we know that there are biotech firms that are investigating DNA and genes and the like. So we're all very familiar with DNA. But at the same time, there's something profoundly mysterious about this molecule that we've never really come to grips with, and that's what my book is about. One way to think of it is to think of something else, another one of the brilliant discoveries of Francis Crick. He was the brilliant English physicist turned biologist. He had been a code breaker in World War II, and he ended up being involved in breaking the ultimate code, the digital code that's stored in the DNA molecule. For 1957, he proposed an amplification of the work that he and Watson had done in 1953, and that was his so-called sequence hypothesis, according to which the four chemicals on the inside of the spine of the DNA molecule, the ones that I've represented with the letters A, C, G, and T, he proposed that those four chemicals, called bases, function exactly like alphabetic characters in a written language or digital characters in a machine code. Now, if you just stop and think about this and set aside the familiarity that we all have with this subject from high school biology, we all have to realize that this is an absolutely stunning hypothesis, one which was confirmed in a series of discoveries over the next 10 to 15 years during this revolution in molecular biology. And what was discovered was this, that these four characters, four chemicals that function as characters, direct the production of proteins and the protein machines that keep the cell alive. The way this works inside the cell is that the arrangements of the A's, C's, G's, and T's they convey instructions for arranging the constituent parts of proteins called amino acids. And here I break for a visual aid. Now, some of you have seen a version of this talk before, so you were getting very frightened by all this amino acid talk, but I'm going to break this down for you. These are snap block beads set on the box from which I stole them, ages 2 to 4. Okay, it's a child's toy, but it illustrates something very important about how molecular biology works. Proteins are like the toolbox in the cell, and they are formed from amino acids, which I've represented with these beads. Proteins have very specific three-dimensional shapes that allow them, in a hand-in-glove fashion, to perform critical functions inside the cell. Just as your toolbox out in the garage may have a hammer and a saw and a wrench and a set of pliers, and each one of those tools has a relationship between its form and its function. The shape of the tool largely determines what kind of a job it can do. The same kind of relationship exists between the shape of the protein and the kind of job that it can do in the cell. And but the shape the protein adopts, and here's some pictures off the internet of some of the intricate three-dimensional shapes that proteins have. Those shapes are determined in large part by the interactions between these amino acids. Each of these amino acids have what are called side chains, little sticky outies off of the backbone there. That's the technical term, the sticky outie. And the interactions between those side chains set in place a constellation of forces that cause different shapes to form, and those shapes in turn, again, are responsible for the functions that proteins can perform inside the cell. Here's an example of a protein, an enzyme protein, that performs it breaks apart a two-part sugar molecule in the cell. You see the cutout in the middle part of the slide there shows the hand-in-glove fit between what's called the active site in the protein and that two-part sugar that looks a bit like a barbell. And you can see that there's a perfect hand-in-glove fit that is responsible for the chemical breaking that the enzyme accomplishes. And this is typical of almost all proteins. They perform their function because of this exquisite three-dimensional specificity, this hand-in-glove fit. Now, the key question in biology is, how does that specificity arise? And we now know that the specificity of shape in the protein derives from the specificity of sequence in the amino acids, which in turn derives from the specificity of sequence, the specific arrangement, the information, the instructions, the digital code stored along the spine of the DNA molecule. So that is what I call the DNA enigma, and I will explain in a minute why I call that an enigma. Before we do that, though, it might be helpful to have a nice visual representation of exactly what the instructions, the digital code in DNA does within the molecule or within the cell. We now know that, and it's nice to have a look. I'm going to, in just a second, press the button, and you're going to see an animation. And it's going to first show you the digital code represented symbolically along the spine of the DNA molecule. And then you're going to see a lot of stuff that starts to happen real fast. And what you'll see is the DNA molecule will be separated by a large protein complex that will open the molecule and expose it for copying. And then you're going to see another large protein complex called a polymerase, which is the cell's copy machine, and it's going to come in and attach itself to the DNA, and then you're going to see that polymerase spit out a single-stranded copy of the DNA information, and that copy is going to go out and eventually direct the production of the protein and protein machines. So I'm going to show this animation now, starting with the digital code on the DNA. Okay, so here we see the DNA and the digital code represented graphically with the A's, C's, G's, and T's along the spine. And now we're going to see this protein complex come and attach itself. And that's preparing the molecule, the DNA strand now, to be copied. And there we're going to see the polymerase attach, and it's beginning to spit out that single-stranded copy, the RNA. This is a view on the inside of the polymerase as individual bases are positioned and added to form this single-stranded copy of the genetic instructions. Now there's the copy completed, and it's going to be transported out to something called the nuclear pore complex, which is a information recognition device that controls the flow of information in and out of the cell's nucleus. Now out it goes here, and it's now going to approach and be transported to a two-part chemical factory called a ribosome. And it's going to thread in there, and as the messenger transcript is read and threads through, it's now going to provide the instructions for building the chain of amino acids, which you see along the bottom there. And a mechanical assembly process ensues as the amino acids are linked up in accord with the three-letter words in the genetic message. Now we see the ribosome, and at the bottom you see the growing protein chain. The spent messenger transcript is at the top. Begins the folding process, and then it's often shepherded out to something called what's a barrel-shaped protein that will cause it to fold with even more specificity. It's called a chaperone. And the animator here in just a second is going to show us when the protein comes into its final conformation. It's going to light up there for us. Boom, there we go. And then door opens, and out it goes to the outer cytoplasm and into the cell where it will do its job. And you can see the animator's done a lovely job himself of bringing to life the beauty and intricacy inside the cell. So that is what we're talking about when we're talking about what DNA does. It directs the assembly instructions. It is the assembly instructions that directs protein synthesis and all the wonderful machines that the proteins make. Okay, so I call this the DNA enigma. But what is the DNA enigma? Well, the DNA enigma is not the question of the structure of DNA. Watson and Crick solved that problem in 1953. In fact, the first line of my book is that when Watson and Crick elucidated the structure of the DNA molecule, they solved one mystery, but they created another. So the mystery is not the mystery of the structure of DNA. It's not even the mystery of where biological information resides. We now know where at least some of the information that living organisms require resides, and it resides along the DNA molecule. The DNA enigma, neither is the DNA enigma the mystery of what the information in DNA does. We now have a very good handle on that. We've seen an animation, albeit perhaps a little simplified, of what's called gene expression or protein synthesis. DNA directs that process. Instead, the DNA enigma is something even more fundamental. It's the mystery of the origin of the information on the DNA molecule. And that mystery is intimately connected with another and a very profound and long-standing mystery in biology, and that is the mystery of the origin of life. Here we have a leading German origin of life biochemist, Bernd Olaf Kuppers, who says that the problem of the origin of life is basically equivalent to the problem of biological information, the origin of biological information. Now, it's fairly easy to see why the problem of the origin of life and the origin of information are so intimately connected. I used to like to ask my students, if you want to give your computer a new function, what do you have to give it? They would immediately understand. They got it. They're better at computers than anyone over 30. And it's code. You have to give the computer you have to give it new code, a new program. The same thing is true in life. If you want to build a new organism from a pre-existing organism, if you want to build a new organ or structure, if you want to build a new molecular machine, the information is required. New proteins are required, and in turn that means new information. More fundamentally, if you want to build life in the first place, if you want to get life going, you need information. The very information that we now know runs the show in biology, the information in DNA. The very first living cell would have required such information, and that is the DNA enigma, the mystery of the origin of the information necessary to produce the first life. Now, for engineers and scientists in the group, one clarifying point might be helpful. There is a mathematical understanding and description of information that was provided by the mathematician Claude Shannon with his, and this is one of his, the simpler forms of one of his equations. And mathematically, information is inversely related to probability. So the more improbable a sequence of characters is, the more information it conveys. There is, however, as Shannon himself pointed out, a limit to that mathematical concept of information. Shannon's equations and his form of analysis cannot help us capture the distinction between these two types of sequences. The sequence on the top is very improbable. It's a very improbable arrangement of characters. Thus, on Shannon's theory, it has a large and calculable amount of information or information carrying capacity. The sequence on the bottom is also very improbable and therefore has a lot of Shannon information, but it has something else, as an extra non-quantitative element. It has a specificity of arrangement that enables that sequence to carry or convey functional information. It performs a communication function. And the term of art in the information sciences is the bottom string is specified information or specified complexity, where the top string is merely complex or it merely has Shannon information. Now, when we talk about the DNA enigma, about the kind of information that is necessary to get life going, it's important to recognize we're talking about specified information, functionally specified information, where the arrangement of the characters matters. And Francis Crick, again, the genius on the cutting edge in the late 1950s, was very clear to clarify this from the beginning of the molecular biological revolution. He said, by information, information means here the precise determination of sequence, either of the bases in the nucleic acid in DNA or RNA, or of the amino acids in the protein. When we talk about biological information, the kind that needs to be explained to understand the origin of life, we're talking about specified, functionally specified information, not merely Shannon information or improbability. Now, I first encountered the DNA enigma at a conference in 1985. There were several scientists there that I've come to know in the ensuing years, but one was a man named Charles Thaxton, who had just published a book called The Mystery of Life's Origin. At this conference, Thaxton shared the critique that he had provided in his book of theories of the chemical origin of life. And he provided an exhaustive critique. And though the conference was set up as a kind of debate between two different schools of thought, nearly everyone on the panel accepted the main thrust of Thaxton's critique, that theories of chemical evolution, as they were called, do not provide an adequate explanation for the origin of the first life. And I became really fascinated with this question because I had been under the impression, having gone through 2 science degrees at that point, that the evolutionary biologists pretty much had all the questions sewn up. And it was rather striking and surprising to me to learn that leading origin of life scientists readily conceded that they did not have, to say not, all the answers or even the key answers to the central question. And I learned that the key question that was a stumbling block for all the theories of the origin of life was the question of the origin of information. So a year or so later, I was off to Cambridge, England, and I began a PhD program after having a number of long conversations with Dr. Thaxton and some of his colleagues about this problem of the origin of life, and I wanted to investigate this mystery. And in my graduate studies, I naturally did 2 things. First of all, I studied the many competing theories of the origin of life that were out there. And one of the reasons that this was such a profound mystery is that the problem of the origin of information is a problem of apparent design. We talked about how the Darwinians attempt to explain away the appearance of design, but when we're talking about digital code, machine code inside a cell, we're talking about something that, as Richard Dawkins himself acknowledges, is uncannily computer-like. Bill Gates, our local hero here in Seattle, has said that DNA is like a computer program, but far more advanced than any we've ever created. And so the question arises, is this appearance of design, this code that looks for all the world, or that functions for all the world like a computer program, or maybe a section of code in a CAD-CAM program, a CAD program in a manufacturing plant, is this the product of intelligence? It looks as if it were designed by an intelligence. Is there any undirected mechanism that can explain away this striking appearance of design at the very foundation of life? That was the question that seized me. And as I began to investigate it, I came across a book by Jacques Monod, a molecular biologist who was a colleague of Crick's in the 1960s. And he wrote a treatise on how scientists should go about solving problems. And on the cover of his book, called Chance and Necessity, Monod said that if you're going to be a scientist, you should approach all problems with the same basic strategies. You should try to explain all phenomena by reference to chance processes, by reference to natural laws, or what you might call necessity, those principles of nature that force the same thing to happen all the time, or by the combination of the two. And as I began to investigate the different theories about the origin of life, I found that sure enough, the various theories exemplified one of these 2 or 3 basic approaches. They either exemplified a reliance upon chance, or a reliance on necessity or natural laws, or what were called self-organizational scenarios, where the laws of physics and chemistry were invoked to try to explain the essential ingredients of life. Or some models attempted to explain the combination of the two. And one such model was called prebiotic natural selection. It tried to invoke Darwin's mechanism of natural selection before life even began, and got natural selection interacting with random events. Now, in my book, I look at a whole series of proposals that have been made to try to solve the DNA enigma, to try to account for the origin of the information that's necessary to build the first life. And I show that each of these proposals exemplifies one of those 3 basic approaches that Jacques Monod very presciently laid out in 1967. And I critique these models, but in some ways it wasn't really my job to critique them. More, I've been able to simply report the critiques that have been made of these models by other origin of life theorists who have become convinced that indeed the field is at a state of impasse. When I give this talk in a longer form, I like to go into a number of these key models and exemplify what's wrong with each of these basic approaches. The good people at C-SPAN, however, have about 65 minutes of tape tonight, so we're going to kind of skate by this, and I'm just going to look at one of these approaches, one that is that kind of exemplifies exactly why the problem of the origin of information is such a deep mystery. And I'm going to look at this approach specifically because it has been one of the most popular approaches to try to solve the problem of the origin of life. It's sometimes known as self-organization. And it relies on what you might call forces of chemical necessity. It was first proposed in the late 1960s by a scientist named Dean Kenyon and his colleague Gary Steinman. And their idea was that you could explain the origin of the specific arrangements of the amino acids that you find in proteins. And I suppose they hoped that you could also explain the origin of the arrangement of the bases in the DNA molecule because of the forces of chemical attraction, forces of necessity. Most of us are familiar with crystals, and in particular, the chemical formula for a crystal of salt, which is, as I recall, Na and Cl. And the Na has a plus charge, the Cl a minus charge, and so there is a force of chemical attraction that creates the structure, an orderly structure, that repeats over and over again. And the idea was that just as the order in a crystal is the product of chemical forces of attraction, so too is the arrangement of the key constituent parts of the biomolecules also the product of forces of attraction. Now, the scientist who advanced this principally was this man, Dean Kenyon. He happened to be at that same conference in 1985 where I heard Charles Thaxton speak, and I learned at that conference that Kenyon was publicly repudiating his own theory, a theory he had called biochemical predestination. For Presbyterians here, he was not a Calvinist. He was referring to the forces of attraction between the constituent parts of these key molecules. In 1985, he publicly repudiated his own theory, which was something of a shock at this conference, sent a ripple through the field of origin of life biology, and it got me even more intrigued with this whole subject. Now, I want to illustrate why this idea of biochemical predestination doesn't work and what Kenyon himself came to in his own understanding of why his theory didn't work. Kenyon realized that even if you had some forces of attraction that could help explain why the amino acids line up the way they do in proteins, you had a more fundamental issue, and that is you had to explain the origin of the information in DNA because we know the DNA produces the sequential arrangement of amino acids in proteins. And as he examined more carefully the chemical structure of the DNA molecule, he realized that his proposal was never going to work. If you look at the chemical structure of DNA, you see on the two outside parts of the molecule little circles with P's and little pentagons. That's the representation of what's called the sugar phosphate backbone. That part of the molecule is the medium upon which the genetic text is inscribed. It is not the information, it's the medium where the information is stored. Along the spine of the DNA molecule, in the vertical axis, where you see the A's, C's, T's, and G's representing the chemical bases, that's the axis where the information is stored. Now, Kenyon's idea, remember, was that you would have forces of attraction that were responsible for causing the key message-bearing, information-bearing constituents of the biomolecules to line up a particular way. Notice that there are no little sticks between the A's, C's, G's, and T's. The little sticks in a structural or in chemistry represent bonds. There are bonds between the G's, the C's, the A's, and the sugar phosphate backbone. But interestingly, those bonds are the same no matter which one of those chemical characters is being attached to the backbone. A lot of chemistry, but let me break it down with another visual aid. Got a little message here, okay? It says mind over matter, mind over matter. It's going to have something about how the Mariners rock, but I'm not sure that they do this yet. So now this is a magnetic chalkboard here, and I've got magnetic letters that stick to the chalkboard. There are forces of attraction here at work. Self-organizational forces, you might consider them, except is the information in this little message the result of the magnetic forces of attraction that cause the letters to stick to the backboard here. Can you see that the information is not the result of the magnetism? Instead, you can see that quickly because the same magnetic forces are at work as I disrupt that message, right? Now, this is a very nice analogy to what's going on in the DNA molecule. The backbone of the molecule corresponds to the backboard here. It's the medium of the message. There are forces of attraction that explain why the bases stick to the backbone, but those forces of attraction do not explain the sequential arrangement of the characters. Do you see that? There are no bonds between the characters, and the same type of chemical bond is responsible for each site where the characters stick. Just as you've got the same force of magnetic attraction no matter which letter you put at exactly this site, okay? So what I'm trying to illustrate here is that there's nothing about the chemistry of the DNA molecule that dictates the sequential arrangement of the characters that constitute the information. It's not the chemistry that produces the sequences that convey the instructions. It's something, as the great physical chemist Michael Polanyi said, it's something extraneous to the physics and chemistry of the system. It would be like saying that the information in this morning's Seattle Times headline is the result of the chemistry of the ink bonding to paper. We know clearly something else is at work. By the way, what else is at work in the... Okay, intuitively, right, we recognize, whether it's my little message on the chalkboard or a newspaper headline, intuitively there's the idea that there is intelligence behind the information. Now, I first encountered this idea in discussions with Charles Thaxton, the chemist I met in 1985, because he had this intuition very strongly. And in an epilogue to his book, he and his co-authors developed this idea, or sketched it out. And when I left for graduate school and began my work in Cambridge, I was seized with a question. Could this intuitive connection between information and intelligence be developed into a rigorous scientific argument? And so naturally, I went back and I studied the scientific methods of reasoning that are used by scientists who are trying to reconstruct the distant past and develop theories of origins. And of course, that led me to Charles Darwin. And it turns out that Darwin pioneered a method of scientific reasoning that goes by a couple of different names. It's sometimes called the method of multiple competing hypotheses. It's sometimes called the method of inferring to the best explanation. And we see here in a quote where he's defending his own scientific method, he says, "It seems to me that supposing that such a hypothesis were to explain such general propositions," he means facts, "we ought, in accordance with the common way of following all sciences, to admit it until some better hypothesis be found out." He's describing his use of this method of inference to the best explanation. And this is the way the method goes. It's designed by scientists who are trying to determine the cause of an ancient event. And they have a limitation. They were historical scientists trying to do this, are not able to go back and actually see the event. So what they do is they propose different hypotheses as to what caused the event, and they evaluate each one in turn, hoping in the best of cases to find only one that is adequate to explain the effect in question. But that leads to a question of scientific method. How do we determine which of the competing causes is best? How do we determine which explanation provides the best explanation for the event that we're trying to explain? Well, it turned out that Darwin and his key scientific mentor, the great geologist Charles Lyell, had a very practical common sense principle of reasoning that they used to guide their scientific method of reasoning about the distant past. And one day, while glancing at the front piece of Lyell's book, I had an epiphany because the subtitle of Lyell's book crystallized this method in a single phrase. He says here, the title of his book is "The Principles of Geology, being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation." The principle was very simple. If you're going to try to explain an event in the remote past, you want to invoke a cause which is known to produce the effect in question. If you want to explain the layer of volcanic ash that exists in eastern Washington, you're going to prefer the volcanic eruption hypothesis over the earthquake hypothesis because we know that volcanoes produce such phenomena and earthquakes do not. So I asked myself a question. What is the cause now in operation that produces digital code? What is the cause that we know of from our uniform and repeated experience? Another part of the methodological dictum of Darwin and Lyell. We should be looking for causes now in operation that we know from our uniform and repeated experience. What is the cause now in operation for the production of information generally? Intelligence. Thank you. It was exactly what I thought. Later, I came across an information scientist, a pioneer in the application of information theory to molecular biologists, who happened to make the same observation. He said the creation of new information is habitually associated with conscious activity. Is that true? Well, indeed it is. Remember our quote from Bill Gates, DNA is like a computer program, but far more advanced than any we ever created. We know that programs come from programmers. We know that information generally, whether we're talking about a hieroglyphic inscription, a section of text in a newspaper headline or book, or a section of digital code in a computer program, always comes from an intelligent source. We know that from our uniform and repeated experience, from our present experience of cause and effect. We know that cause that is now in operation. So as I examined the competing approaches to explaining the DNA enigma, the origin of the information necessary to produce the first life, I very consciously developed the case for intelligent design using the very method that Darwin himself had used, the method of multiple competing hypotheses. And in the book, I go through this in a very systematic way, and I look first at hypotheses based on chance, those based on necessity, and those based on the combination of the two, and show from the literature in the area of origin of life biology that each of these approaches have failed. And instead, there is only one known cause of the origin of information, and that cause is intelligent design, conscious activity. And therefore, if there's only one known cause of the given effect, the presence of the effect points strongly back to the action of the cause. And so that's the argument that I've developed in Signature in the Cell. That's, in a sense, the argument based on Biology 101. One of the things that's also very exciting, which I address in the book, and I'll only briefly touch on here in closing, is that there are many other signatures or fingerprints of intelligence in the informational properties of living systems. My colleagues at the Biologic Institute, the lab that we've started here in Seattle and in Redmond, have been working on a computational simulation of that animation that you saw there, of what's called gene expression. And one of my colleagues is a computer programmer. He was an architect-level programmer at Microsoft, and he was working closely with our molecular biologist. One day he came into my office. He dropped a book on the desk in front of me. It was called Design Patterns. It's apparently a standard manual for software engineers. And my colleague said to me, the programmer, he said, "I'm getting an eerie feeling that someone figured all this out before us." And I said, "What do you mean?" And he explained to me the concept of a design pattern. Is apparently a term of art in computer science. It means a design strategy or a design logic. And he said there's all kinds of different design strategies, design patterns that we use to process information. And I'm finding all of them inside the cell, or many of them. And he said the interesting thing is that they are and they started talking Microsoft talk. It's like they're an 8.0, 9.0, 10.0 version of the same strategy that we use. And he said they're just executed, same design logic, but executed with much more engineering elegance. And some of the things, and I discussed some of these design patterns in the last part of my book, the fact that we have messages within messages in the genome. It's called nested coding of information. There's a files within folders, folders within superfolders way of storing and organizing information, a hierarchical filing system in the genome. There is a distributed storage and retrieval system for genetic modules, for informational modules, informational data sets. And the operating system in the genome, which we used to think was junk DNA, by the way, it's not junk. The operating system causes those modules to be accessed and concatenated, and that's what we now understand a gene to be. At every level, we're seeing a hierarchy of information, And it's just an absolutely fascinating system that's been being revealed by the most current discoveries in modern biology, in modern genomics. So it's a very exciting time to be interested in molecular biology and genomics, but there's all of the new studies, all the new discoveries are showing that the case for intelligent design is even stronger. Each one of these design patterns is a feature for which there is only one known cause in the universe, and that cause is intelligent design. Now, in closing, I'd like to make one more point, because a common objection to intelligent design is that it's an argument from ignorance, that we're arguing from what we don't know rather than what we do know. My sometimes debating partner, Michael Shermer, has made this objection repeatedly. He says intelligent design argues that life is too specifically complex to have evolved by natural forces. Therefore, life must have been created by an intelligent designer. He's accusing us of arguing in a fallacious manner like this. He's saying that the argument for design is basically simply constitutes nothing more than our ignorance of what natural processes can do. He's saying we're arguing natural processes cannot produce the effect in question. Therefore, since we can't think of anything else, we invoke the mysterious notion of intelligent design. But in fact, the notion of intelligent design is not mysterious. It's something that we know about from our own repeated experience. And the argument is not an argument from ignorance. It is an argument from what we know, both about the features of the cell and the genome, but also about the cause and effect structure of the world. So the argument for intelligent design actually goes like this. None of the natural processes that we've examined, not chance, necessity, or the combination of the two, have demonstrated the power to produce the effect in question, the specified information that runs the show in biology. But we do know of a cause, a type of cause, which is known to produce that effect. That cause is intelligence. Therefore, intelligent design constitutes the best explanation based on what we know from biology and from our knowledge of the cause and effect structure of the world. That's the form of argument that Darwin used. If this is an unscientific or fallacious argument, then so was his. But instead, what I've done is actually turn tables and show that by using Darwin's method and applying it to information, to evidence that he did not yet know about, we can now show that the central legacy of Darwin is not that there is no evidence of design, but rather we can use Darwin's very method to reaffirm the case for design based on these very exciting discoveries of modern biology. And I thank you.